Okay, so let's start off with the fact that viruses suck and that they like to steal genetic information from their hosts. And here, this is a paper in Cell, so very high impact, where this group has described that viruses are able, and they're describing a mechanism by which viruses, or at least a subset of viruses, will steal genetic information from their hosts and possibly create chimeric proteins with both virus components and human components. So what could be a possible strategy for this? Well, one that I can think of is that when a virus is wanting to take over a host, it's probably beneficial to have components that come from that host because it makes it more difficult for the host's immune system to recognize those viruses. So if you could imagine the perfect virus or the perfect virus or the perfect pathogen is going to be one that uses proteins that are within your own body so that it becomes more and more difficult to create antibodies against those viruses. Because the moment you make antibodies against the virus, those are the same antibodies that are going to attack you as a person. Okay? So here, this group has described one of the ways that a subset of viruses known as segmented negative-stranded viruses, negative-stranded RNA viruses, this is the mechanism by which they can cause chimeric proteins. Now, granted, this is not to say that this is the only mechanism by which viruses can employ to make chimeric proteins, but they're saying that this is one of the mechanisms that they are employing. So, with this, let's go through the name is hybrid gene origination creates human virus chimeric proteins during the infection. And so here they have a graphical abstract, which I love. I love graphical abstracts, and I wonder if Cell actually requires that in their for their papers. So here we have the 5' end. So just so you know, DNA and RNA, when it's written, is written from the 5' end to the 3' end. So just keep that in your mind. That is, it has something to do with the carbon numbering of the ribose or the deoxyribose sugars that make up DNA or RNA. Just know that the 5' end refers to the beginning of the virus, or beginning of the genetic material. So here we have the 5' end of host mRNA. So mRNA is short for messenger RNA. So when you have DNA, you have the DNA, which is the genetic code of a organism, usually. When this DNA needs to be converted in quotations, it gets converted into its protein product. The first step is the transcription from DNA into RNA. And so, or I should explicitly say DNA gets transcribed into messenger RNA first. And then that messenger RNA, a portion of it gets translated into the final protein product, usually. So here, we're starting off with, this is the host messenger RNA. So this would be the messenger RNA for any one of the proteins that are expressed within the cell. So all of the cells in your body are producing proteins at all moments of time. So because of that, there's going to be a plethora of messenger RNA that is filling the cells that are getting ready to be translated into the final product. So the DNA gets transcribed into messenger RNA, and you have a bunch of that floating around in the cell. Okay, and so this would be one of them. And when there is an infection, and this is specifically influenza, IAV, and I forget the exact, or the word be acronym, but it's IAV for influenza. And this will have an enzyme, or find a way, I don't know if it's an enzyme yet, to slice or cut off a piece of that host mRNA. Okay, one of the many. And it probably does it for more than one. And then you have the cap of the mRNA that it has a little bit of extra mRNA on it. And the benefit of this is that this little extra bit of mRNA from the host can anneal, it can pair up with the viral RNA. Okay, so let's keep this in mind. The viral RNA is negative stranded, and we'll go into more detail about that later, but it can complement this, this part of the cap of the host mRNA. And then when this happens, then eventually there could be a polymerase or an enzyme that extends or duplicates RNA that binds to this area and then initiates and creates the complementary strand to the virus RNA. So this virus RNA is originally negative stranded, which means it is not the strand that has the genes. It is the strand that complements the strand that has the genes. So remember, with all of your DNA, complements each other, and so one strand, at least in humans, there might be a few exceptions, and I would get back to you if those pop out to me. One strand will have the genetic material for the actual gene. The other is just the complement, right? So two peas in a pod, they come together. Only one side is actually going to have the gene. Some viruses, I believe, don't quote me on this, can actually have genes on both sides, but that's getting a little bit too far ahead. In this particular case, the virus RNA that is in the original virus is not the genetic material that encodes for DNA. So its first thing that it needs to do is get duplicated or complementarily duplicated. And one of the ways to initiate that is to line up with some bits of messenger RNA on the human, or at least from the host. So after it comes in and the complementary strand gets annealed or attached to this cap, you end up with the generation of a chimeric host virus mRNA. Okay, so just know that a lot of viruses employ this strategy to come up with the final mRNA version. And so this is the known mechanism. You will have this cap, which came from the original mRNA, and then you have this untranslated region that is at the beginning, and then you have this area that actually encodes for the gene. And so in all eukaryotes that I know, this starts with AUG. This is the adenine, uracil, guanosine codon. So if we can remember, the translation machinery will read 3 letters of RNA at a time to produce an amino acid. So it'll read 3 letters, produce an amino acid, then read 3 more letters, and then attach another amino acid, and then read 3 more letters and attach a different amino acid. And it continues until it reaches a stop codon. So, and there are 3 different stop codons normally. So here we start with AUG, and normally this produces the canonical or the normal virus protein. Okay, but this might not be the case. So sometimes what might actually happen is when in the process of getting this cap, sometimes the cap will have the AUG start earlier than it should. And I shouldn't say should, but let's say the virus normally wanted this to happen, where its first protein or its first amino acid is the methionine, the AUG. Rather than it being there, the host actually had one earlier. And so when the translation happens, oh crap, you're not starting here where you were supposed to start. You're starting all the way back there. And so what can happen is you start adding start adding parts of the messenger RNA, or so I should say, you start adding parts of the protein that was a part of the host organism's genetic material, and you are appending that to the viral protein. And so here, if it's in frame, and by in frame, it means that you're lucky enough that, remember how the translation machinery reads 3 amino acids at a time? Well, sometimes you can shift those 1 or 2, and then you have frame shift problems that go on. So, but if it is in frame, you end up having the canonical virus protein with a little bit more of the human, or I should say the host peptide attached to it. And so this would be a completely different, I shouldn't say completely, this would be a different enzyme or a different protein. And if that causes an advantage or an evolutionary advantage for the virus, then the virus, this particular mutation might end up being conserved later. So this would be one way that it could happen. A different way would be this production, where it's an off-frame mutation, or I shouldn't say mutation, but the reading frame gets shifted. And so rather than producing the normal virus protein, it produces a completely different virus protein that may or may not have any function at all. So it does that in conjunction with a part of its host protein as well. So, and in this case, they're calling it an UFO. It's absolutely alien. It's an upstream Frankenstein open reading frame, and they just called it a UFO. And you know, in biology, we love our acronyms, especially if they make sense to even a moderate degree. And so whichever acronyms pass the, what would the word be? Some acronyms are better than others, and then some of them just stick, and so we basically evolve acronyms as well. So continuing, this is the graphical abstract of what they're describing in this paper. So the highlights. So here they are describing a mechanism of hybrid gene birth, and it is employed by many families of RNA viruses. Human RNA and viral RNA encode new genes together. Hybrid genes either make extensions of viral protein, or they make completely novel proteins, and these are called UFOs, which is what I described here. So human virus genes and proteins play a role in pathogenesis and are conserved. So if this human virus gene is conserved, if it provides an evolutionary advantage to this single virus particle that is being produced, then that single virus particle is going to be more successful at duplicating within that human host, and then it is more likely to spread to fellow humans. Okay, so let us continue. So here in eukaryotes, ribosomes typically recognize mRNAs with a terminal 5' cap structure. And the 5' just means that it's at the beginning of the RNA, so just keep that in mind, which is followed by an untranslated region. So this is called the UTR, and this can be tens to hundreds of nucleotides in length. So a lot to unpack there. So the ribosome is the protein, or the riboprotein machinery, that will turn, or I should say translate, mRNA into its final, or I shouldn't say final, but into its protein product. So ribosomes will recognize these mRNAs with the terminal 5' cap. So this terminal 5' cap is a requirement for the ribosome to function correctly or to find the mRNA correctly. So let's say you're this alien virus and you just inject your RNA into a human cell. That might not necessarily be sufficient for the ribosome to produce the virus proteins. So what do the viruses do? The viruses can steal these caps from the mRNA that is already within the cell. So let's continue. Oh, and the untranslated region. So you have this cap that is on, let me go to the graphical illustration. You have this cap that is on the mRNA, and there could actually be a significant period of, what would you call it? A lot of nucleotides, which are the letters of DNA and RNA. You could have a lot of letters that are not transcribed or translated at all. And then once you get to this AUG, or this start codon, which also happens to encode for methionine, this is where the translation starts. And so this is where the protein starts. You will have, some DNA and RNA that just does not get translated at all. And here they call it the untranslated region. So continuing, however, a growing body of work has shown that translation can initiate in the 5' untranslated regions of a large proportion of eukaryotic mRNAs, and sometimes extremely close to the 5' cap, resulting in upstream open reading frames. So every once in a while, even within a healthy cell, there might be the opportunity for these ribosomes to start translation a little bit early, or very early, which could extend the protein further back. So here, they're saying a large subphylum of RNA viruses, and these are the segmented negative strand RNA viruses, or the SNSVs. And segmented just means that it has, segments. It has, for lack of a better term, it's almost like chromosomes. So you know how you have chromosomes, or 23 pairs of chromosomes that are not connected to each other. They, in of themselves, are separate. Some viruses also have that as well. So let's see if, like going onto Wikipedia. So a negative sense single-strand RNA virus is a virus that uses negative sense, and negative sense means the nature of the roles of the strand and its complement in specifying a sequence of amino acids. So positive sense, this is not a good, positive sense means that the DNA, or the genetic sequence, is going to directly be read by the ribosomes to turn itself into a protein later. That's positive sense. Negative sense means that it first needs to be the complementary strand, or the opposite strand of that negative sense strand, needs to be produced first, and then that positive strand is then what makes the final proteins. And so this only, you really only care about this when it is a single-stranded virus, or a single-stranded organism, but I guess viruses are the only ones that you worry about. So, and in this particular case, it's dealing with RNA. So, and RNA and DNA can both have the instructions of genetic code within them. And so there are some DNA viruses, there are some double-stranded and single-stranded DNA viruses, there are some single-stranded and double-stranded RNA viruses, and the single-stranded ones are the ones where you need to consider either the negative sense or the positive sense. Because if it's double-stranded, then you have both the negative sense and the positive sense all within one virus. So let's see. Single-stranded RNA viruses are classified as positive or negative depending on the sense or polarity of the RNA, and the negative viral RNA is complementary to the mRNA and must be converted to a positive RNA by RNA polymerase before translation. So RNA polymerase would be the polymerizing enzyme that will make the final strand. So going back to here, the RNA polymerase is what is going to take this structure and then start extending or creating the complementary strand of the virus's RNA. So let's continue. I've been rambling a little bit. So these segmented negative strand RNA viruses, or SNSVs, include families such as the Rnaviridae, the Peribunaviridae, and the Orthomyxoviridae. So highly contagious human and animal viruses like influenza A virus, so IAV, and Lassa virus, or LASV, belong to these families and are responsible for significant levels of morbidity and mortality worldwide. So in segmented negative strand RNA or RNA viruses, viral mRNA synthesis is primed using short 5' methyl 7 guanosine capped RNA sequences. And should I go into this? I will a little bit. So let us just Google search this. So when you are, we'll just call it look at the 5' cap. When your DNA gets translated into RNA, one of the ways that it gets processed is through this 5' cap. And I'll just quickly go over it. So in eukaryotes, this 5' cap is found in the 5' end of an mRNA molecule, and it consists of a guanine, which, remember, guanine is one of the letters that you can find in DNA and RNA. So guanine, it consists of a guanine nucleotide connected to an mRNA through an unusual 5' to 5' triphosphate linkage. So this would be the best way to describe it. So normally, DNA gets, if we have our what would you call it? Ribophospho backbone. So here you have ribose, here you have a phosphate, here you have a ribose and another phosphate, and you have this chain, this polymer. This is the 5' carbon, and then this is the 3' carbon. So here we go 5' to 3', and so whenever you hear a 5' and 3', just know that means you're starting with the 5' carbon on this ribose, and you're going in the direction of the 3' of the ribose. That's the best way I can describe it. So normally it's 5' to 3'. Just keep that in mind. And then the cap is this 7-methylguanosine. So guanosine would be normally like this, but they add this methyl group onto it, and that causes a positive charge on this guanosine. And rather than being bound 5' to 3', it's actually going to be 5' to 5'. And so this is the 5' to 5' triphosphate bridge, okay? And this is just one of the things that your body does to help identify what mRNAs are of you and what mRNAs are of completely different organisms. So knowing this, we'll keep on going. So knowing this, viruses, learning that they need to have this 5' cap to have efficient translation, knowing this, they have found a way to steal that cap from the host. And so you could make the argument, maybe there's an organism that does not use this 5' methyl 7 guanosine cap. Maybe there's a completely different organism that doesn't use that. Well, a virus, maybe it uses a completely different cap. A virus, if it infects a different organism, it's going to want to steal whatever cap they use. So perhaps I don't know if this is true or not. Perhaps it's another organism that a virus could infect, and if it infected that different organism, then it wouldn't matter if they used the 5' methyl 7 guanosine cap or not. It would steal whatever the host is using, and still be able to initiate translation. So, rambling. Let's continue. So in segmented uh, uh, segmented negative strand viruses, viral mRNA synthesis is primed using short 5' methyl 7 guanosine capped RNA sequences, which the viral polymerase cleaves from host RNA polymerase 2 transcripts in a process known as cap snatching. So basically, the viral polymerase is able to steal it from the host's RNA polymerase. So I would assume, I'm not going to go too into it, I would assume the host RNA is copying this from DNA, or I should say transcribing it from DNA, and then somehow the RNA polymerase of the virus steals it from the host in some way. So we'll just keep on going with that. We'll just keep on going. So cap snatching creates viral transcripts that are genetic hybrids of hosts and viral sequences, with the host-derived 5' sequences being highly diverse. So why is it highly diverse? Because you have a diverse set of mRNAs within each of your cells. So it doesn't matter if it's from the same host or not. All of your cells are going to have a lot of different mRNAs with caps on them. And so the benefit for a virus is that it gets this huge repertoire of different nucleotide sequences that it can steal from. And if it steals from any of those caps, then it ends up producing a wide variety of different chimeric mRNAs. Now, I shouldn't say most, but canonically, what those chimeric mRNAs are supposed to do is normally you just ignore the blue part and you make the same virus protein no matter what. That would be the known canonical viral protein synthesis mechanism. But there is the opportunity, with this mechanism, there is the opportunity for the chimeric protein, since there is so much variety in it, to still have this, to start earlier. And if it accidentally starts earlier, then you will have all of this being made instead. You will get part human or part host protein, and you will get part virus protein. And that is just the reality of what could possibly happen. So, oops, did not mean to do that. So let us continue. So cap snatching creates viral transcripts that are genetic hybrids of host and viral sequences, with the host-derived 5' sequences being highly diverse. So once made, viral mRNAs are translated by the host machinery. So in this manuscript, we hypothesize that appropriating 5' terminal mRNA sequences from their hosts, that the segmented negative strand viruses could obtain functional upstream start codons, or AUGs, which is a mechanism we termed start snatching. So they're not just cap snatching, they're also start snatching, which the start codon is AUG. It normally encodes for the methionine amino acid, but if you have if a virus accidentally gets a piece of the virus, or a piece of the host mRNA that has this upstream AUG, it might completely screw up or completely change things downstream of that upstream start codon. So translation from the host-derived upstream start codons in chimeric host viral transcripts would access upstream viral open reading frames, also known as upstream viral or UVORFs, depending on the frame of the upstream AUG relative to that of the canonical viral protein. So here they're just saying, and that is in relation to this this photo here, is this normally untranslated region, how do I phrase that? So normally, you could have the canonical virus that is attached to both this untranslated region and the human or the host protein, or it could just be completely different if it's off frame or if there's a shift some way. And normally one would assume that it would actually not be good to just have what would be almost a completely random amino acid sequence, but we would assume that if this is utilized by viruses throughout the course of their life cycle, I guess that would be the wrong term from it, but as viruses are growing and evolving over the course of their existence, then we would presume that if they are utilizing this strategy, then the normal reading frame and the shifted reading frames would probably both create functional proteins of some sort. So there are some viruses that actually employ the strategy to compact more different proteins into the same genetic sequence, just shifted by a few. But that's not always the case. Not all viruses employ that strategy. So let's continue. So the results. The influenza A virus cap snatches sequences that contain upstream AUGs. So influenza A virus gene transcription is initiated by cap snatching from a host mRNA. This process generates IAV mRNA with a 5' portion derived from the host. This mechanism is used to express viral genes that encode canonical viral proteins. So we hypothesized that start codons, or AUGs, within host sequences could generate upstream host virus chimeric open reading frames with coding potential. So depending on the reading frame, a host-derived upstream start codon might initiate the synthesis of two novel chimeric genes encoding for an N-terminally extended viral protein. And N-terminally, so remember when we said the genetic material goes from the 5' to the 3'? When it comes to protein sequences, we normally say it starts from the N-terminal side and ends with the C-terminal side. And N stands for protein, and N stands for the amino or the nitrogen side, and C stands for the carboxylic acid side of the protein. So just know that the N-terminal side means the beginning section of the translated protein. So depending on the reading frame, a host-derived upstream AUG might initiate the synthesis of two novel chimeric genes, which encoding for an N-terminally extended protein. So let's continue. Or alternatively, an entirely novel protein overprinted on the canonical viral open reading frame. So let's go to this figure. So here they're describing the process. Host mRNA, you get the cap anneals onto the virus's RNA, and here they have the virus's RNA exactly matching with this. But what could also possibly happen is there might be a little bit of mismatch, and also this cap might possibly, rather than having this virus perfectly get to the very end, it could be that this virus curls back a little bit because the matching isn't exactly on the end of the virus's RNA. That I'm just spitballing right now, but that could also happen as well. And that might be a way that you could remove some of the virus's RNA as well. Continuing, you end up with this viral mRNA, and here this is outcome 1, which is the known mechanism. You have your untranslated region of the virus, and none of the host mRNA gets translated. And you start normally and you just produce the canonical viral protein. But here's the outcome number 2. If you have the normal frame, you would create the viral protein with the human encoded extension. So here's the human encoded extension, and then here is the virus portion of that protein, all fused together. If this AUG, sorry, focus, if this start codon upstream does not line up perfectly, because remember we read 3 nucleotides at a time, if this does not line up perfectly, then you might shift everything and just completely jumble up the entire message. But sometimes you might still encode for a functional protein, miraculously almost. So that would be the other option. So these outcomes are contingent on 2 assumptions: that the upstream start codons are present in the capsnatch host sequence and can enable translation initiation. So sometimes you can have a start codon, but the surrounding genetic material isn't conducive at allowing the ribosome machinery to attach to it. So it's possible that you could have a start codon, but it can't be read at that location, and so it starts off at a downstream area. What is happening here is saying upstream start codons can be present in the capsnatch host mRNA sequence and can enable translation. So they're saying both. Sometimes both can happen. And the 5' mRNA transcribed from the virus's untranslated region should lack stop codons. So here, there could be a mechanism where there is a stop codon in the untranslated region of this virus protein or this virus mRNA normally. And so you could view these stop codons as breaks that would completely prevent this entire thing from happening. So if you had this AUG here, and if there was a stop codon here, it would completely eradicate, or it would just make small peptides rather than creating the virus protein. And so one could make the argument that if a virus took the cap, if a virus really wanted to protect one of its proteins because it needed to be conserved, well, maybe put a few stop codons in this untranslated region, and that would prevent this chimeric mutation, if you will, from ever happening, or happening, what would the word be? Commonly. It wouldn't allow it to happen commonly. So let us continue. These outcomes are, no, I read that already. So furthermore, the absence of stop codons interrupting the untranslated regions or the downstream open reading frames should be evolutionarily conserved. So let's see. So if viruses use this strategy frequently to mutate faster and to basically accelerate evolution, then they would prefer to not have these stop codons in the untranslated region. So if this was a strategy that caused evolutionary advantage to the viruses as a whole, we would expect to see a lot of, or at least a stop codon in these untranslated regions, which would prevent this entire thing from happening in the first place. So, to address the first point, we determined the abundance of the upstream start codons in capsnatch host sequences archived in a decap and 5' end sequencing, or this is called defend-seq dataset, that we had previously generated from these A549 cells, which were let's look up what A549 cells are. I believe these are just these are adenocarcinomic human alveolar basal epithelial cells. So they are a cancer cell that is cultured for studies in just human experiments. They're just cultured, and it's nice to have these cells because cells multiply forever, or I should say, these cancer cells will multiply forever, and we treat them almost as if they are, bacteria. But the nice thing about them is that if we want to run tests on human cells, it's easier if we have cancer cells rather than having to do these experiments on humans, because to do these experiments on humans would be pretty unethical. So if we work with the cancer cells that are growing in our test tubes, that is a way to do a lot of these biomechanical experiments. So, and these are cancer cells that are from the alveolar, the alveolar epithelial cells. So they did that with the, how do I phrase that? They had previously generated from the A549 cells infected with the influenza virus, which was H1N1. And so the start codon containing host-derived cap sequences ranged from 7 to 20 nucleotides with a median length of 11 nucleotides. So similar to the distribution obtained for all cap-snatched sequences. So host-derived oligonucleotides with the AUG codons were present at similar ratios in all 8 genome sequences of the virus and were present in all 3 reading frames, constituting 12% or about 12% of all cap-snatched sequences. So about 12% of, So about 12% of the cap-snatched sequences had an AUG. If I'm reading this correctly, maybe I'm not. So AUG containing host-derived cap sequences range from 7 to 12 nucleotides with a median length of 11 nucleotides, and similar to the distribution obtained for all cap-snatch sequences. Let's continue reading. Maybe I'll learn more as this goes. I didn't actually read this paper before we started, so I'm learning at the same rate as you are. So host-derived oligonucleotides with AUG codons were present at similar ratios in all 8 genome segments. Okay, so the genome segments are the segments of the virus RNA. So influenza, I believe, has 8 segments. So influenza segments. Yeah, so influenza has 8 segments associated with it. So, oops, I lost my spot. Here we go. And so host-derived oligonucleotides with AUG codons were present at similar ratios in all 8 segments of the virus and were present in all 3 reading frames, constituting about 12% of all cap-snatched sequences. So here, that I think is actually very interesting. So as these viruses are randomly getting caps, about 12% of them equally distributed among all of the segments of the virus's RNA, about 12% of them have these early start codons in them. So that's pretty interesting. And it's not just the perfect reading frame where the start codon gets on and then you have the extension and then the proper translation of the normal virus protein. The other two, where they are shifted 1 or 2 in the wrong direction, or I should say off frame, those are also very present. So that is interesting. Granted, that might not necessarily translate into the final protein in the same ratios, because the start codon might not necessarily be in the right location or they might not have good surrounding nucleotides that would allow translation to happen properly. But they're saying in about 12% of these mRNAs that they find, they, among all 8 segments of the influenza A virus, do you have early start codons? Interesting. So, host-derived oligonucleotides with AUG codons are present at similar ratios in all 8 genome segments of the virus. I read that. Similar results were also obtained when we performed cap analysis of gene expression, also known as CAGE, on primary human monocyte-derived macrophages infected with different strains of influenza A virus. So these results indicate, upon infection, neither the virus nor the host cells appear to prevent the formation of chimeric RNAs with hybrid coding potential. So they're just saying that there appears to not be a mechanism that is effective at preventing these chimeric RNAs from forming with the AUGs. So we would presume that after billions of years of these viruses existing, I shouldn't say billions, but hundreds of millions of years of these viruses existing with their current hosts, or with the host and similar species of that host, we would presume that if this was detrimental to create these chimeric mRNAs, that they would have found a way by now to prevent these chimeric RNAs from happening. And this would be important if these chimeric mRNAs were getting in the way of proper evolution. But it appears as though no. It appears as though neither the host nor the virus have an effective way at preventing this from happening. So I mean, if 12% of the segments have this happen, makes sense to me. So we performed a bioinformatic analysis to determine if stop codons were absent from the influenza A virus sequences within the 5' untranslated regions, and if so, whether this was evolutionarily conserved across influenza A virus strains. So first, we analyzed the nucleotide sequence variability of the 5' untranslated regions of all 8 segments using all influenza A virus H1N1 strains available from the NCBI influenza virus database. So here I'm just going to make a note. Let's say they did find stop codons in the untranslated region of some segments. This could tell you that those particular segments might be more protected from this chimeric process. So here we have our untranslated region of the virus proteins, and this would be the normal start of those virus proteins. If there was a segment that had stop codons in this untranslated region, that might help protect the final protein from being that might protect this protein from being chimerized, for lack of a better term. So we'll just keep on going for now. So we found, we were talking about the database. I should really stop clicking to prevent. So the 5' untranslated regions of each individual segment are highly conserved within each individual segment. So there's good conservation of the untranslated regions, which is surprising. So as shown by the positional weight matrices and sequence alignment, we then translated the 5' untranslated region of each genome segment in silico, which means with computer simulations in all possible frames. This revealed that the 5' untranslated region of every influenza genome segment can maintain a reading frame in at least one frame. Okay, so we found that the 5' untranslated regions of 5 out of 8 genome segments lacked upstream stop codons in frame with major open reading frames. So an upper panel. Okay, we'll go. Actually, we'll go to it. Figure 2. You know what? I'm probably going to have to spend another hour looking at those figures without being recorded to fully understand them. So these segments thus have the potential to code for N-terminally extended viral proteins. Stop codons were, so 5 out of the 8 genome segments lacked upstream codons in frame with the major open reading frame. So 5 out of the 8 did not have a stop codon that were in the same frame, or at least within the same. 5 out of the 8 would not be able to undergo this chimeric protein mechanism. Why? Because if the start codon were to start and then be in frame with the rest of the normal virus protein to produce this, normally there would be a stop codon in the way that prevents it. Granted, that is not to say that this other off-frame mutation, which creates these UFOs, that could still be an option in those in the 3 that do have stop codons in the normal frame. Granted, that might be a little bit too much to hold in your head at the moment. So these segments thus have the potential to code for N-terminally extended viral proteins. So stop codons were also absent from the 5' untranslated regions of 6 of the 8 genome segments when these were read out of frame. So, 6 of the 8 genome sequences did not have any stop codons for out-of-frame viruses. So 6 out of the 8 could have theoretically gone through this mechanism where you have the start codons from the host organism, and then you just create these UFOs, is what they're calling them. So so there would be 5 of those 8 could undergo this sequence, and 6 out of the 8 can undergo this sequence of events. Okay, so interesting. This suggests the intriguing possibility that in the presence of a host-donated start codon, these genome segments could make novel genes and encoding hybrid polypeptides. So, and polypeptides are proteins. So to probe the length of the upstream viral open reading frames, we translated viral sequences that had cap-snatched upstream start codons in our dataset in computer simulations. The result of these analyses indicated the general propensity to create chimeric ORFs with half of the viral genome. So, genome segments predicted to make sizable products or greater than 30 amino acids. So these ORFs overlap with canonical viral genes, but are read in different frames. They range from over 40 residues to nearly 80 residues, where N-terminal extensions of the major open reading frame were possible, and these range from 8 to 21 amino acids in length. So thus, upstream viral open reading frames are present in all genome sequences, and if licensed by host-derived upstream start codon containing RNAs, this could generate polypeptides of varying lengths. So, yeah, let's keep on going. So host virus mRNA chimeras associated with elongating ribosomes. So when cap-snatched host upstream start codons, if cap-snatched host upstream start codons did initiate translation of viral 5' untranslated regions, the 5' termini viral mRNAs would be bound by initiating ribosomes. We therefore performed ribosomal profiling of influenza virus infected cells in the presence of Harringtonite, and this particular mineral blocks elongation of newly assembled ADS initiation complexes, but not of those already engaged in elongation. So, how do I phrase that? By adding this mineral, which is Harringtonite, this will stop the elongation of newly assembled ribosomes. It'll prevent new ribosomes from latching onto the mRNA, but those that are already engaged in elongation, those will continue to do what they want to do. So ribosome protected fragments, or RPFs, were mapped to both the human and viral genomes. Mapping of RPF sequences revealed an accumulation of ribosomes at the canonical initiation site of mRNAs transcribed from all 8 genome segments, consistent with previous reports, as well as observing ribosomes accumulating at the canonical initiation site. We also observed RPFs mapping to the host-derived sequences upstream of the 5' untranslated regions, suggesting that translation initiated in this region. So this is saying that they are getting hints that the start codons in these 5' that these start codons that are upstream of the untranslated region, those are also getting the ribosomes to latch onto them and initiate translation. So you are not just producing the canonical viral RNA or canonical viral proteins, you are also possibly initiating the production of peptides as well. So this is broadly consistent with the proportion of cap-snatch sequences containing upstream start codons. So focus, come on. So, you know what I'm going to do is, this is a little too much to go through all at once. I'm just going to skip to the discussion now. So, the discussion. In this manuscript, we describe the existence of a mechanism employed by these segmented negative-stranded viruses to generate chimeric host virus genes. This mechanism, called start snatching, involves the co-opting of start codons from host mRNA sequences to expand the viral proteome. This mechanism appears to be accessible to all SNSVs, including major human pathogens such as IAV and LASV. Start snatching allows the translation of proteins from cryptic upstream viral open reading frames, either as canonical viral proteins with the N-terminal extensions, or as UFO proteins overprinted in the canonical viral ORF in open reading frame. In this study, we have identified examples of both types of UV-ORFs in the influenza infections. We have shown that translation can initiate on upstream AUGs in the host-derived sequences of viral mRNAs, and that this leads to the expression of chimeric host virus proteins that can be detected in infected cells. So, in our hands, the ablation of the UV-ORFs did not impact viral replication in vitro, but had moderate effects in vivo, which would be consistent with upstream viral ORFs encoding accessory proteins. So we found that UV-ORFs can be recognized by the immune system, and we modeled the contribution of different evolutionary forces at play on UV-ORFs by characterizing viral intrinsic and host immune features that contribute to their evolution. Finally, we showed experimentally and by sequence analysis that the capability to express UV-ORFs through start snatching is widespread among the SNSVs. So, we hypothesized that cap snatching of SNSVs could generate ORFs that are encoded by 2 genomes, the human and the virus. Consistent with this, our analyses indicate that roughly 10%, that's not insignificant, 10% of the influenza A virus mRNA contains host-derived upstream AUGs. Furthermore, UV-ORFs are translated in at least 3 of the 8 IAV genome sequence segments, generating NP, EXT, PB2 UFOs, and PB1 UFOs. So this is the N-terminal. These are the extended ones that have extra host, what would you call it, peptides added, and then you have the PB2 UFOs and the PB1 UFOs, which are when you have the code shift. So genetic evidence suggested that many other UV-ORF proteins are also likely to be expressed, although we did not detect them in our current studies. So potentially due to the substantial sequence overlaps of the N-terminal extensions with canonical viral proteins and the short lengths of many overprinted ORFs. Overall, our analysis indicates that multiple families of viruses can generate chimeric RNAs and could produce proteins through this mechanism. So, you know what, I'm not going to go over this. What I am going to go over is this is not necessarily talking about how the viruses can snatch host RNA and then I would say present that or allow that to, what would the word be passed on to their progeny. That's not what this might not necessarily be the case, but what they are saying is that given one virus that infects one cell, about 10% of these proteins are going to be, or I should say 10% of these mRNAs are going to have some chimeric quality to them, because about 12% of all the segments have one of these things happening to them. So, yeah, I guess what I'm going to end with is this would, if this was evolutionarily conserved, we would expect there to be at least a suppression of mutations that take place in these untranslated regions. So we would normally assume if you have these untranslated regions of viruses, we would assume that if you have mutations in those areas, they might not be conserved very easily because they're not even encoding for a protein in the first place. But if there is a subset of the virus protein that should, in fact, have these untranslated regions be translated, then you really could have some conservation among it, especially if there is conservation among different reading frame shifts that take place. And granted, I'm getting a little bit too off topic here. So, so keep in mind, this is a quality or a strategy that is employed by the negative-stranded RNAs. Can positive-stranded RNAs employ a similar mechanism in some way? Possibly. It hasn't been described yet, but I could see there might be a possibility, especially if you get infected with 2 viruses at the same time. Let's say you got infected with one of these let's say you got infected with one of these negative-stranded, single-stranded, or segmented negative-stranded RNA viruses. At the same time, you got infected with a positive-stranded RNA virus like the coronaviruses. Then it is possible that you end up creating a chimera with the yeah, I mean, you could make a chimera between the virus RNA. You can make chimeras with the viruses and the host RNA. I mean, there's a laundry list of different things that could happen. Granted, that would be contingent on getting both viruses within the exact same cell, which would be less likely, but probably possible, right? So with that, I'm going to head on out. I just want everyone to be aware of this paper, and if you want, you can read this paper yourself. But yeah, I think it's interesting. So thanks for listening, and hopefully I will see you tomorrow.